Welcome back to The Deep Dive. Today, we are undertaking a truly adventurous mission, diving into a comprehensive paper by independent researcher Alfred, titled Coupling Mechanisms Between Human Bioelectric Fields and Dark Matter Plasma.
Mm-hmm.
This paper, it doesn't just review bioelectricity, it takes what we can currently measure, inside every single cell, and attempts to bridge that classical biological study with some really out there concepts in modern physics.
Absolutely. We're talking about dark matter here.
Exactly, the existence and nature of dark matter.
It's quite a synthesis of fields, really. Alfred's hypothesis, it goes way beyond the standard models of, say, how nerves fire or muscles contract.
Right.
He's proposing that these weak, observable bioelectric fields, the ones we rely on for physical development and function, are actually interpenetrated by something else.
Something invisible.
An invisible, highly structured, low-energy dark plasma sheath. That's the term he uses. And in this model, this sheath acts as a kind of scaffolding.
A scaffold.
Yeah, linking our ordinary body to a much larger, non-ordinary dark plasma body. And the suggestion is that this potentially enhances our computational and memory capabilities during life.
Okay. That demands a massive shift in perspective, and honestly, a healthy dose of skepticism as we go through this.
Oh, absolutely. Critical thinking is key here.
So the initial question we want you, the listener, to hold on to throughout this deep dive is this. If the electrical blueprint of life, the one that controls our form, which we can measure with instruments, what if that's only half the picture? What does that invisible, energetic second half look like? And how is it supposed to be built? We're going to unpack this framework step by step.
Right.
Starting with the absolute foundation, the electric currents that define life in the first place. Okay, let's start at the microscopic level, where the rubber meets the road, really, the cell membrane.
Right.
To generate a body-wide electrical field, the body has to create charge separation somehow. So what are the mechanisms? How does this happen inside every one of our cells?
Well, it's an act of constant biological effort. Every living cell maintains a boundary, right? The membrane. It separates the inside from the outside.
Okay.
And to achieve charge separation, the cell uses specialized machinery. Think of them like tiny gates and pumps, ion channels, pumps, and transporters.
Oh, okay.
And their job is to meticulously control the concentration of charged particles, specifically ions, things like sodium, potassium, calcium, chlorions.
These are the charged atoms.
Exactly. By moving these charged particles unequally across that membrane boundary, the cell actively generates what's called an electrochemical gradient.
Electrochemical gradient. That sounds important.
It's a critical term. It means there's both an electrical difference, a voltage, and a chemical concentration difference across the membrane. Two kinds of imbalance working together.
And the most famous piece of machinery doing this work is the sodium-potassium pump, right? How does that specific process create this essential voltage?
The sodium-potassium pump. It's like the tireless worker bee of the cell membrane. It uses energy, the cell's fuel, adenosine triphosphate, or ATP, to actively transport 3 positively charged sodium ions out of the cell for every 2 positively charged potassium ions it brings in.
So 3 positives out, 2 positives in.
Exactly. If you look at the net result of that exchange, more positive charge is leaving the cell than entering it.
Ah, right.
And this persistent differential movement happening constantly generates a net negative charge inside the cell relative to the outside. That establishes the resting membrane potential, the baseline voltage.
And this isn't just passive leaking, it's active work the cell is doing.
Absolutely active transport. It's constantly working to maintain that gradient against the natural tendency for things to even out.
Okay, so we have this constant flow of ionic current, but the paper emphasizes something important here. We shouldn't just think of these as generic electrical charges moving around.
That's a key point Alfred makes. When we talk about these ionic currents, particularly the movement of hydrogen ions, which are just protons, or even potassium ions, we are essentially talking about the movement of protons.
Protons themselves.
Fundamentally, yes. So the currents generated are essentially proton currents. And this has enormous implications, according to the paper. This uneven distribution of charged particles, these proton currents, flowing across membranes and through the fluid spaces in our tissues. It's effectively a non-neutral proton near plasma that is intimately linked to and generated by the biochemical body.
A near plasma, what does that mean exactly?
It means it's a localized soup of charged particles, mostly protons in this context. It's not a fully independent, self-contained plasma like you'd find in space or a fusion reactor.
Right.
but it shares some characteristics.
Hmm.
It's this charged environment, this electrical engine, that's really driving everything at the cellular level.
And this voltage difference created by this proton near plasma, it's absolutely necessary not just for sending signals, but for the basic integrity and function of the cell itself.
Oh, it serves multiple vital purposes.
Yeah.
That voltage difference is crucial for maintaining osmotic balance, basically controlling where water goes, keeping the cell from swelling or shrinking.
Okay.
It provides the electrical excitability necessary for tissues like nerves and muscles. It allows for instantaneous communication across distances.
Right, like nerve impulses.
Exactly. It's the basis for complex cell signaling pathways that control everything from gene expression to coordinating cellular behavior. We see this very dramatically in the nervous system.
Yeah, so.
Well, the rapid, precisely controlled opening and closing of voltage-gated ion channels. These are channels that only open when the voltage across the membrane hits a certain threshold.
Ah, voltage gated.
Yes. That rapid change creates the action potential. That's the electrical spike that travels down a nerve fiber, allowing signals to traverse the nervous system and ultimately trigger things like muscle contractions. It's all based on managing that voltage. #tag 1.2 living electric cells.
Okay, so if every single cell is engaged in this active transport, constantly moving charges, generating voltage, the sheer volume of this activity across the entire body must be staggering.
It really is.
How should we conceptualize the human body in electrical terms then?
We really need to move beyond thinking of the body as just a collection of chemicals in bags. The paper suggests thinking of the body as an enormous integrated electrical system. An electrical system, yes. Every one of the roughly 40 trillion cells in an adult human body, it's essentially a tiny, rechargeable proton battery or capacitor.
40 trillion batteries.
Think about it. They all possess these ion channels. Skin cells, bone cells, muscle cells, nerve cells, constantly managing these electrical proton currents. This coordinated system ensures that the body's tissues aren't electrically neutral. They possess a measurable, consistent voltage.
And this voltage isn't just some kind of static background noise, it's dynamic, especially during development, right, when the organism is first forming.
This is a really key insight that Alfred highlights. Stem cells, those fundamental, undifferentiated cells that haven't yet specialized, they actually start out with zero voltage.
Zero, really?
Zero resting potential. They only acquire their necessary negative voltage as they mature and commit to becoming a specific type of cell, like differentiating to a neuron or a muscle cell or a fat cell.
Wow.
This actively demonstrates that the electrical component isn't just an effect of being alive. It seems to be a critical, fundamental driver in cellular differentiation and the whole process of development. The electrical signature appears before the final functional form.
That's fascinating. And we can actually put numbers on this resting voltage for different tissues in an adult.
We can, yes. The voltage varies depending on the cell's specific job and its metabolic activity. Resting nerve cells, for instance, typically maintain a potential of approximately -70 millivolts. -70.
Okay.
Musculoskeletal tissues, because they need to be highly excitable and have a particular structure, they tend to rest around -90 millivolts. Even fat cells maintain a potential, usually resting around -50 millivolts.
So everything has its own electrical set point.
Exactly. This constant electrical state, maintained by that controlled flow of proton currents through those voltage-gated ion channels, that's the baseline. This proton near plasma, constrained by the cell structures and visible to our instruments, this is the ordinary matter electrical foundation that Alfred believes is somehow coupling with something much more subtle, something non-ordinary.
All right. The ordinary electrical foundation. Let's move from there. So we have trillions of cells, each one a tiny battery, all working together, creating these voltages. Let's zoom out now. Does all this collective electrical activity actually manifest as a cohesive, organized field that, surrounds and maybe even guides the physical body?
Well, the history of the research suggests exactly that, and it goes back surprisingly far, nearly a century ago.
Really? Who was looking into this?
It starts with a researcher named Harold Saxton Burr. He was a professor of anatomy at Yale University, and his work in the 1930s and 40s was really quite revolutionary for its time.
Okay, Burr at Yale, what did he do?
Burr was convinced that electric fields played a fundamental organizing role in life. He developed methods to measure bioelectrical fields in developing organisms, things like plant seedlings and salamander embryos, using extremely sensitive voltmeters.
And what did he find?
Well, this was the startling discovery. He found that the electrical fields he measured did not simply reflect the current, small, developing size or shape of the organism.
They didn't match the physical form.
Not the form it had at that moment. Instead, The field mapped the finished product.
The field was the future form, like a template.
Precisely. Burr found that the bioelectrical field surrounding these tiny seedlings and salamander embryos was already shaped like the eventual adult form, and it occupied roughly the same volume that the adult would eventually fill.
Wow. So the electrical pattern, or blueprint, as he called it, wasn't just latent potential, it was already there, measurable.
Exactly. He even measured this kind of field pattern in an unfertilized egg. Burr concluded that these voltage gradients were organized into specific, persistent patterns that lasted throughout the entire life of the organism. And, very importantly, he could measure these fields outside or above the physical body.
Outside the body.
Yes. He essentially argued that all living things are molded and maintained by these underlying electrodynamic fields.
That idea, this invisible, persistent blueprint, it sounds like it's bordering on metaphysics almost, yet you're saying modern science has actually given us ways to see it now. Hashtag 2.2, visualizing the electrical shadow.
Absolutely. The work of researchers like Michael Levin and his colleagues, working much more recently on bioelectricity, uses highly advanced techniques to visualize exactly what Burr could only measure as subtle voltage gradients.
How do they visualize it?
They use something called a voltage reporter dye technique. It's quite clever. They can introduce this special dye into an embryo. They famously did this with developing tadpoles.
Sure.
This dye has the property that it converts the electrical potential of the cell membranes into varying degrees of brightness, essentially fluorescence. So the resulting image is basically an electric map of the developing organism.
A map of the voltage across its surface. And what did that map reveal in the tadpole experiment?
It was really compelling. The map showed this vivid pre-pattern of the future frog's facial features.
Before they were physically there.
Yes. Before the physical matter, the cells had begun to coalesce into the recognizable structures of a face. The researchers could see an electrical outline, a faint but perfectly structured little mouth, a nose, an eye socket, the jaw structure, all laid out electrically.
And this appeared before the physical parts.
Hours, even a full day, before the features physically materialized. The electrical pattern came first.
So the visual electrical pattern is like the instruction set, and the physical cells just follow it. They fill in the lines?
That's exactly the interpretation.
Yeah.
Science writer Sally Adee actually coined a great phrase for this, the electrical shadow.
Electrical shadow. I like that.
It describes this phenomenon perfectly, a projection of the future state of development, just as Burr had suggested decades earlier. And crucially, this electrical shadow wasn't confined just to where the cells were. It seemed to extend slightly beyond the developing visible body. And when the physical body did form hours later, the features, the mouth, the eyes, they coalesced exactly where that electrical pre-pattern had dictated they should be. It really looks like a master control system.
This persistence, the fact that the pattern is there first, it raises a philosophical question maybe, but one grounded in these findings that Alfred's paper really picks up on. If this blueprint persists throughout life, like Burr claimed, and if we could somehow use that voltage dye technique on, say, an adult human, what would we actually see?
Well, theoretically, you'd see the constant electrical scaffolding of the adult form. And if that scaffolding is persistent, if the adult mold stays constant, even as the ordinary matter body, ages, regenerates, cells turn over, maybe even gets injured,
Right.
it suggests the existence of a high-fidelity, persistent template, one that's somehow maintained or updated, perhaps independent of the constant biological turnover of the physical cells themselves.
So this measurable, organized, persistent electrical blueprint, that's the definitive starting point as we move into the next, maybe more challenging part of the paper.
Exactly. It sets the stage for observations that seem to go beyond what a standard voltmeter can tell us.
Okay, this is where we make a critical jump. And frankly, this is where we need to apply some real critical rigor. We've established that science can measure and visualize an electrical shadow that guides biological form.
Right. The blueprint exists.
Now, Alfred's paper pivots. It argues that certain individuals, people he terms sensitives, claim to be able to see this electrical shadow, but, and this is the key part, they see it with far more structure, detail, and associated matter than any scientific instrument can currently detect. So the obvious question is, why should we, as, scientists and critical thinkers, even entertain these subjective reports?
That is absolutely the right question to ask. And Alfred's rationale for entertaining them isn't that they constitute proof in themselves.
Okay.
But rather is because the core claim, the existence of a structural pre-pattern guiding development, was actually made by these sensitives decades, even a century, before Burr and Levin experimentally confirmed aspects of it.
So the historical precedents.
Exactly. Alfred uses these subjective reports not as proof of the mechanism he proposes, but as an observational data set, a set of observations about persistence, structure, and perceived density that requires some kind of physical model to explain it. He's essentially trying to provide a potential physics-based framework for phenomena that have historically just been dismissed as purely anecdotal or hallucinatory.
Okay, so he's trying to explain the observations, not necessarily validate the observer's interpretations wholesale. Hashtag tag 3.1, the historical claim of non-ordinary perception. Let's review those historical claims then, the ones that predate the modern bioelectricity research. What specifically did these early 20th century sensitives describe seeing?
Well, we can look at records from experimental metaphysicists, people like Charles Leadbeater, writing around, say, 1910.
Okay.
He reported observing, using what he called non-ordinary sight, something he described as a doll-like little figure hovering about and afterwards within the body of the mother.
A doll-like figure, not the physical fetus.
Explicitly not the physical fetus, but described as a structural mold or template for it. Another researcher, Arthur Powell, elaborated on this around the same time, describing this figure as accurately reflecting the shape and size of the infant body it was in the process of building.
So a template matching the final form.
Yes, but here's another crucial detail they noted. This mold appeared to develop faster than the ordinary matter fetus that was linked to it.
Faster. So a time-resolved mold that's ahead of the physical growth. That sounds remarkably like Levin's pre-pattern showing up hours before the features.
Precisely. From Alfred's perspective, what Leadbeater and Powell were describing sounds very much like a time-resolved electrical shadow, a dynamic mold that seems to run ahead, maybe even accelerating the developmental rate of the physical body it's associated with.
And these accounts were published 50 to 100 years before Burr and Levin provided experimental evidence for these electrical pre-patterns.
Exactly. Which, for Alfred, makes them compelling, if still anecdotal, descriptions of something real, perhaps a non-ordinary matter component associated with the blueprint. This leads him to hypothesize. Maybe certain people have a biological predisposition, perhaps genetic, or maybe it can be trained, an ability not just to vaguely sense electric fields, but to perceive the actual matter structure that's attracted to and organized by those fields.
That idea connects to something Michael Levin observed in his work too, doesn't it? The idea that the cells themselves access some kind of bioelectric memories.
Yes, Levin noted in a 2014 paper, I believe, that the cellular collective seems to be performing what he called mind-boggling computations by accessing these bioelectric memories that dictate the target morphology, the final shape.
Mind-boggling computations.
Alfred links this directly to observations reported by Barbara Brennan. She was formerly a NASA atmospheric physicist who later focused on energy healing and perception.
Okay, Brennan, what did she report seeing?
Brennan reported seeing, using her non-ordinary perception, various complex three-dimensional pre-patterns being actively projected, seemingly by the genome itself, as if the genome was calculating and projecting specific electrical blueprints with different intensities or strengths.
So not just one blueprint, but maybe alternatives.
Potentially. And interestingly, Levin's subsequent work has indeed confirmed the existence of these alternative bioelectric pre-patterns. He views this as the cellular population, computationally solving the problem of deciding what morphology to create, especially if development is disrupted.
So the computation Levin talks about, Alfred suggests, might be happening partly in this perceived non-ordinary structure. Hashtag tag 3.2 evidence from beyond the body. Okay, the idea that this structure isn't just energy, but actual matter that persists even beyond the ordinary physical body. That's reinforced by some other observations, isn't it? Like with plants or amputations.
This is quite compelling, yes. Brennan specifically reported observing an energy field matrix, a structured pattern in the exact organized shape of a leaf being projected by a plant before the physical leaf had actually grown into that space.
The blueprint again.
Yes. And even more dramatically, she reported that if a fresh, living leaf was physically cut, the energy field, the blueprint of the entire whole leaf, remained visible for some period afterwards.
Even though part of the physical leaf was gone.
Exactly. This strongly suggests that the structural matrix, the blueprint, has some independence from the ordinary matter components it organizes.
And we hear similar claims from people reporting near-death experiences, right? Seeing phantom limbs.
Yes, that's a common report. Individuals during near-death experiences frequently describe seeing their own body, or sometimes other people, with limbs that have been physically amputated, appearing fully present and complete in this non-ordinary or energetic field.
Which would imply the complete adult pattern, the blueprint, persists somehow, even when the ordinary matter component is removed or damaged.
Precisely. And if that's the case, it means there must be some form of persistent, invisible matter occupying those spaces, matter which our current scientific instruments simply fail to detect.
The paper also brings in the accounts of Robert Monroe. He did extensive self-experimentation involving out-of-body experiences, which he termed being in his second body. What did he report seeing that connects to electricity specifically?
Monroe's accounts are detailed. He reported that while operating in this state, which Alfred hypothesizes is perhaps the lowest energy layer of a larger dark plasma body, his relationship with ordinary electromagnetic fields was fundamentally different.
How so?
He described being able to clearly see bright structural electrical field lines, like the ones radiating from high-voltage power lines, or even the detailed structure of a Faraday cage, which is designed to block electric fields.
But electric field lines are normally invisible to us.
Completely invisible to ordinary human sight. So Monroe seeing them strongly implies that some form of otherwise invisible matter was being physically attracted to and concentrated by these measurable electric fields, making them visible to his non-ordinary perception.
And since Monroe's second body itself was invisible to ordinary observers,
Alfred posits that both the field lines Monroe perceived and the non-ordinary body he was operating within might be composed of this same invisible or dark matter, matter that interacts with strong electromagnetic fields. Hashtag 3.3 experiments on bioelectric fields versus dark bioplasma.
okay, to try and bring this back towards some hard data, the paper relies quite heavily on a series of controlled experiments conducted by Valerie Hunt at UCLA back in the 1980s and 90s.
Yes, Hunt's work is pivotal for Alfred's argument.
Her approach was interesting because she used scientific instruments and human sensitives simultaneously, trying to find a correlation. Can you describe her setup?
Hunt's methodology was quite sophisticated for the time. She placed her subjects inside electromagnetically shielded rooms. These rooms allowed her to precisely manipulate the atmospheric electrical and magnetic environments independently.
A controlled environment.
And crucially, she used electrodes placed on the subject's skin to record physical electromagnetic data frequencies, wave patterns, field strength, while an aura reader, one of these sensitives, was simultaneously present in the room but isolated to independently record their visual observations of the subject's energy field or aura.
Two parallel data streams, instrument readings and visual reports. What happened when the electrical environment was manipulated? This seems like a critical test of how sensitive our biological field is to external electrical forces.
It was dramatic. When the electrical component of the shielded room's atmosphere was significantly withdrawn, basically reducing the external electrical field towards zero, the effects on the subjects were immediate and quite profound.
What did they experience?
Their measured energy fields, the readings from the electrodes, became scattered, disorganized, incoherent. And the subjects themselves experienced profound sensory impairment. They reported losing awareness of their body boundaries. They couldn't tell where their arms or legs were in space without looking.
Disorientation.
Yes, and often accompanied by emotional distress. Some subjects would burst into tears, reporting feelings of threat or profound uneasiness, even though logically they knew they were safe in the room and weren't actually sad about anything.
Wow. So how does Alfred interpret that finding?
Alfred interprets this, and Hunt's own conclusions support this view, as evidence that the ambient electromagnetic environment we live in acts as a kind of confinement mechanism for our physical and perhaps energetic body. Removing that external field leads to systemic disorganization, a loss of coherence.
That's a staggering idea that our basic sense of self and location is so tightly coupled to the external electric field. What about the magnetic field? What happened when that was changed?
That produced different but equally striking effects. When the magnetic field intensity was isolated and then dramatically decreased, the subject's motor coordination instantly deteriorated.
Motor skills.
Yes, they struggled with simple balance tests, couldn't stand on one foot. Fine rotor skills, like the classic finger-to-nose test, became difficult or impossible.
Okay. And when the magnetic field was increased?
Conversely, when the magnetic field was increased beyond the normal ambient state, their coordination didn't just return to normal, it improved dramatically, sometimes beyond their usual capabilities.
Better than normal.
Subjects could stand easily on one foot, even on tiptoes for extended periods. They could lean at seemingly impossible angles without losing balance. It suggested the magnetic field strongly influences the neural pathways related to balance and motor control, perhaps by affecting the flow or coherence of the electrical blueprint itself.
Fascinating. Now let's get to what might be the most crucial point from Hunt's experiments for Alfred's thesis, the correlation between the electronic measurements and the aura reader's visual observations.
This is really the bridge that Alfred relies on to connect the measurable biofield to the perceived structure. Hunt found that the aura reader's visual observations, descriptions of colors, shapes, patterns, locations in the energy field, showed an incredibly consistent and reliable correlation with the electronic measurements taken by the electrodes at the exact same location on the body.
How consistent?
For instance, if the sensitive consistently observed the color blue in the energy field around the subject's shoulder, the electrodes placed on that shoulder would always record the exact same specific frequency and wave patterns associated with blue in their system. This wasn't a one-off. It was repeated across multiple sensitives, observing different subjects, confirming a strong, reliable link, a kind of sympathetic resonance between the measurable bioelectric patterns and the visually perceived aura or energy field.
So while the sensitive sees color and intricate structure, the instrument sees corresponding measurable frequencies and waveforms. This really suggests a physical entity is being perceived, not just imagined.
It strongly suggests that what the sensitives perceive is not random noise or hallucination. It's highly correlated with the body's actual measurable bioelectric state. And this correlation apparently extends to clinical applications as well.
How so?
The paper cites the work of Shafika Karagula, a medical doctor and psychiatrist who collaborated with sensitives. They found that these individuals could often identify underlying medical problems like tumors, infections, or obstructions by observing specific distortions, breaks, or areas of stagnation in the energy patterns of patients, often before physical symptoms were obvious.
Which aligns perfectly with Burr's earlier findings about field disturbances, and also Levin's note about cancer cells often showing anomalous zero voltage, a break in the electrical blueprint.
Exactly. So the picture that emerges is that sensitives are perceiving information-rich detail, color, texture, structure, flow within an energy field that is organized by the underlying electrical blueprint that instruments measure. But they're seeing aspects of it, perhaps its material structure, that our standard instruments cannot yet fully record or resolve.
Which forces us back to the question, what is this stuff, this structured energy field that seems to have substance that sensitives report seeing? Okay, so the gap seems clear now. On one hand, we have a measurable bioelectric field, these proton currents creating an invisible blueprint that science can detect.
Right.
On the other hand, we have these consistent reports from sensitives observing a persistent, detailed, three-dimensional, almost plasma-like matrix, something that seems to possess its own form of matter.
And this is what drives the need for a new model, beyond just the standard bioelectric field.
Barbara Brennan, the former NASA physicist, described this field, didn't she, as something interpenetrating the physical body.
Yes, she described it as looking like a sparkling web of light beams, a matrix that interpenetrates the physical body. She concluded quite firmly that the current scientific model, based solely on biochemistry and standard electromagnetism, was insufficient to explain it. This matrix, she theorized, serves as the primary persistent pattern or template upon which the physical tissues are shaped and maintained. She was essentially saying the blueprint isn't just disembodied electrical energy, it's a structured form of subtle matter itself. Hashtag, tag, 4.1 dark bioelectric field, and the need for a new model.
And what did Brennan hypothesize about the composition of this human energy field she observed? What did she think it was made of?
Well, based on her observations of its behavior, she described it as having fluid-like motion, similar to air or water currents, with eddies and flows. She concluded that this human energy field is likely composed of extremely tiny particles, possibly subatomic, operating in the state of a biological plasma.
Biological plasma. We should probably give some context for plasma here.
Good idea. Plasma is often called the fourth state of matter, after solid, liquid, and gas. Sometimes the fifth state, depending on how you count. Essentially, it's a cloud of charged particles, typically ions, atoms that have lost or gained electrons, and free electrons. The key thing is that the particles are energized enough and spaced far enough apart that they aren't just bouncing off each other like gas molecules. Instead, their behavior is governed primarily by long-range, collective electromagnetic forces. They act as a collective fluid.
Okay, so a cloud of charged particles behaving collectively. This brings us to the first critical challenge for J. Alfred's paper, doesn't it?
It does.
If sensitives are potentially seeing some kind of bioplasma, could it simply be an ordinary bioplasma body? made of the standard particles we already know about, the protons and electrons generated by our cells.
This is exactly the hurdle the paper has to overcome before it can justify leaping to dark matter. Alfred meticulously reviews this ordinary bioplasma hypothesis specifically to demonstrate why, based on known physics, it fails to account for the kind of structure, persistence, and containment that's being reported. Tag 4.2. Is there an ordinary bioplasma body?
Critical examination. Okay, let's review that ordinary bioplasma theory. It was proposed before, wasn't it? Primarily in Eastern European research circles back in the 1970s.
That's right. Researchers like Inyushin and Sedak proposed the existence of bioplasma, composed of ordinary free electrons and ions, perhaps concentrated mainly in the highly metabolic areas like the brain and spinal cord. They thought it could be generated by processes like those occurring in the mitochondria, the powerhouses of the cell.
And what was their main evidence for this?
Well, a lot of their core supporting evidence at the time came from analyzing Kirlian photography.
Ah, Kirlian photography. Those images showing colored coronas or flares around objects like fingertips or leaves when exposed to a high-voltage electric field.
Exactly. They interpreted the variations in these Kirlian discharges of the streamer color, length, intensity as directly visualizing this bioplasma body. However,
There were problems with that interpretation.
yes, by 1976, subsequent more rigorous research pretty conclusively demonstrated that most, if not all, of the variations seen in Kirlian photographs could be largely explained by mundane environmental factors. Chiefly, the moisture content and even microscopic dirt on the surface of the object being photographed. Higher moisture meant a brighter, bigger corona.
So it was mostly just electrical discharge interacting with surface moisture, not revealing an independent plasma body.
That became the scientific consensus, yes. It severely undermined the idea that Kirlian photography was a reliable window onto some hidden bioplasma structure.
Okay, so the Kirlian evidence is weak. But even setting that aside, Alfred argues there are fundamental, insurmountable physical challenges to the idea of a substantial, permanent, ordinary bioplasma body existing inside or around a warm, wet, ordinary human body. Let's break down those three detailed arguments he makes.
Right, the paper is very clear on this. These three challenges really demonstrate why a stable, permanent, ordinary bioplasma body is physically impossible under normal biological conditions. Challenge number 1, temperature requirements. Forming ordinary plasma requires intense energy. You need to hit atoms hard enough to strip electrons away and create that cloud of charged particles. For common gases to become stable plasma, the temperatures required typically range from 4,000 up to 10,000 Kelvin.
That's thousands of degrees Celsius, hotter than the surface of the sun in some cases.
Exactly. The biomolecular tissue of the human body, operating at a cozy 310 Kelvin, roughly 37 Celsius or 98.6 Fahrenheit, would be instantly vaporized or scorched by contact with plasma that hot.
Okay, so hot plasma is out. What about cold plasma? That exists, doesn't it?
Cold plasma does exist, yes. But creating and maintaining it requires extremely strong containment fields, usually powerful electromagnetic fields generated externally in a lab, which leads directly to this second challenge.
Right, challenge number 2, electromagnetic strength, or rather the lack of it in the body.
Precisely. The electrical fields naturally generated within the ordinary human body are incredibly weak compared to the external fields needed to generate or contain cold plasma in a laboratory setting. Furthermore, the ordinary electric charge itself, the fundamental force between electrons and protons, is actually very strong.
Strong charge, weak field. Why is that a problem?
Because when charged particles are moving relatively slowly, as they would be in a cold biological environment, that strong attraction between opposite charges, positive ions, and negative electrons leads to extremely rapid recombination. They find each other and neutralize almost instantly.
Like static electricity dissipating in humid air.
Exactly like that. The extremely weak endogenous fields in the body are completely insufficient to overcome this strong attraction and prevent this rapid recombination. This makes a stable, permanent, large-volume ordinary plasma body highly unstable. It would just dissipate back into neutral gas or liquid particles almost immediately. No persistence.
Okay, so the strong charge coupled with the weak field means no stability over time. And the third challenge deals with the nature of the charged particles. We do have those proton soups, the neuroplasma we started with.
Challenge 3. Hydrodynamic versus electromagnetic control. Yes, we do have concentrations of charged particles in the body, like those proton currents flowing across cell membranes, but the paper argues that these concentrations are generally non-neutral, over large spaces, more positive here, more negative there. And crucially, they are generated and constrained primarily by hydrodynamic processes.
Hydrodynamic, meaning fluid dynamics.
Yes, fluid dynamics and
Oh.
mechanical pumping actions, all constrained and directed by the physical, biomolecular structures of the cell, the membranes, the channels, the pumps. For a concentration of charged particles to qualify as a true independent plasma body, according to physics definitions, the collection of particles must be overall electrically neutral, equal positive and negative charges, and its behavior must be governed primarily by long-range electromagnetic effects, not just fluid mechanics. A true plasma can exhibit self-organizing features like vortexes, filaments, and double layers, independent of its physical container.
But our proton soups can't do that.
Our proton soups are structurally constrained by the cells and tissues, they're often non-neutral, and their flow is dictated by the biochemistry. They cannot collectively constitute a separate, independent bioplasma body capable of this kind of complex, self-organized electromagnetic behavior. They are a consequence of the biology, not an independent entity coexisting with it.
Okay, that is a pretty convincing case against the ordinary matter hypothesis then. The basic physics of ordinary matter, temperature, charge strength, field strength, it just doesn't seem to allow for a persistent, organized, unconstrained plasma body to exist at biological temperatures and conditions.
Precisely.
Mm.
So, Alfred concludes, if the visual observations made by sensitives, reports of structure, persistence, color, non-local computation, are reflecting something real,
Yeah.
and if ordinary plasma is physically ruled out as the explanation, then the structure they are perceiving must be composed of a different, more subtle form of matter,
Which necessitates the jump.
which necessitates the jump into dark matter physics. That's the logical next step Alfred takes in the paper.
Okay, this is really the core of the deep dive then. Since ordinary physics seems to prohibit the kind of persistent, structured plasma body that's being described, Alfred theorizes that the ordinary bioelectric field is actually interpenetrated and augmented by self-interacting dark matter plasma,
Right.
a hypothesized, stable subcomponent of dark matter itself.
This is where we really move into highly specialized theoretical physics. The standard picture of dark matter, as you know,
Yeah.
is that it interacts primarily, perhaps only, through gravity. It's dark because it doesn't seem to interact with light, with ordinary electromagnetism.
Right, that's the challenge.
Alfred is proposing that a subset of this dark matter might not be completely inert. It might possess its own form of charge, its own dark electromagnetism, and crucially, that it can be stable enough to form a complex plasma structure, even at relatively low biological temperatures. The paper then outlines the unique proposed properties of this dark plasma that allow it to overcome those three critical challenges that ruled out its ordinary counterpart. #5-5.1, self-interacting dark matter plasma.
Okay, so what are these unique properties? What allows this hypothetical dark plasma to be stable and long-lived in a biological environment, unlike ordinary plasma?
There are two main proposed properties, flowing from theoretical physics models about a hidden or dark sector that could confer this stability, assuming dark matter possesses its own form of electromagnetism separate from ours.
Property number one.
The first is the concept of a weak, dark electric charge. Many standard physics models exploring physics beyond the standard model hypothesize a hidden sector with its own forces and particles. This often includes a theoretical new long-range force, sometimes called dark electromagnetism.
Like our electromagnetism, but just for dark matter.
Exactly. Mediated by its own force carrier, perhaps the dark photon, associated with what physicists call a U1 gauge field, basically. A force field analogous to our own electromagnetic field, but one that primarily or exclusively couples only to dark matter particles.
Okay, but what about the charge strength? That was key for ordinary plasma instability.
Right. The crucial proposed property is the strength of the charge associated with this dark force. The dark electric charge is theorized to be far, far weaker than the ordinary electric charge carried by electrons and protons. Estimates vary, but it could be anywhere from 1/100th down to perhaps even 1/1,000,000th of the strength of the ordinary electric charge.
Wait, hold on. A charge that's potentially 1,000,000 times weaker, wouldn't that be far too weak to organize anything useful, especially in biology? We usually think of strong forces being needed to structure matter.
That's a really important question, and it seems counterintuitive at first. Yes, it's weaker, but remember, even 1/1,000,000th of the electric force is still incredibly strong compared to gravity something like 10 times stronger, so it's plenty strong enough to organize matter. But the relative weakness compared to ordinary electric charge is actually the key advantage for stability in a cold, dense environment.
How so?
Because the fundamental charge is so much weaker, The mutual attraction between oppositely charged dark particles, dark electrons, and dark protons is greatly suppressed. This inherent weakness fundamentally prevents that rapid, chaotic particle recombination that plagues ordinary plasma at low temperatures.
Ah, the recombination problem.
Exactly. The weak charge allows charge separation to be maintained much more easily, even with weak confining fields. And maintaining charge separation is absolutely essential for storing information, performing computations, or building any kind of stable, complex biological structure. The weakness enables longevity.
Okay, so the weakness paradoxically allows for stability. What's the second stabilizing factor proposed?
The second factor relies on the predicted composition of the dark sector itself. The presence of a neutral particle buffer. Cosmological observations and simulations suggest that the dark sector likely contains significantly more neutral dark matter particles than charged dark matter particles, perhaps a ratio of around 5.5 neutral particles for every charged particle pair.
More neutral stuff than charged stuff in the dark sector? How does that help stabilize the plasma?
These abundant neutral dark matter particles would act as a kind of surrounding insulator or buffer for the charged dark particles. They effectively increase the average physical distance between the charged dark particles, dark electrons, and dark ions, and they also reduce the frequency of all non-electrostatic interactions, like simple physical collisions.
So they keep the charged particles apart and reduce random jostling.
Precisely. This neutral buffer further dampens potential instabilities and helps ensure that the dark plasma remains coherent, structured, and stable over very long periods. It perfectly fulfills the theoretical need for a permanent, organized matrix that could coexist with biology.
And critically, because dark matter, by definition, interacts so weakly with our ordinary matter.
There's no threat to the biochemical body.
Yeah.
Exactly. The inherently low-energy, weakly interacting nature of this proposed dark plasma means it could potentially interpenetrate our ordinary tissues without burning them, vaporizing them, or chemically disrupting them in any significant way. It neatly sidesteps the temperature and reactivity problems that made ordinary plasma impossible in this context.
Okay, so we have a theoretical mechanism for a stable, invisible dark plasma, but how does it physically know where our ordinary bioelectric field is? If it interacts so weakly, what's the glue? What confines it to the electrical blueprint generated by our ordinary cells?
Right, you need a coupling mechanism. The paper argues that simply having stable dark plasma isn't enough. It needs to be attracted to and organized by the ordinary bioelectric field. To achieve this, Alfred invokes the hypothesis of a new, short-ranged, attractive fifth fundamental force.
A fifth force, beyond gravity, electromagnetism, and the strong and weak nuclear forces.
Exactly. A new force that specifically mediates interactions between ordinary matter particles and dark matter particles. But this force can't be universal like gravity, or it would have been detected easily already. It must be highly selective in what it interacts with to avoid messing up all the known physics of atomic nuclei and chemistry.
Selective interaction, what kind of selectivity is proposed?
The proposed selectivity is absolutely key to making the model work without contradicting known physics. This hypothetical fifth force is theorized to be strongly attracted to ordinary electrons. It's also thought to be moderately attracted to ordinary neutrons. But, and this is crucial, it's proposed to be hardly attracted to, or potentially even slightly repelling, ordinary protons.
Okay, why that specific pattern? Strongly attracted to electrons, moderately to neutrons, but basically ignoring protons. What's the rationale?
This specific selectivity seems engineered, you might say, to achieve two critical goals simultaneously. First, the force must couple strongly to the electrons in our cell membranes and atoms, because the behavior and distribution of these electrons are what generate our macroscopic bioelectric fields. The force needs to see the electrical blueprint.
Makes sense. Attract to the source of the field.
Second, the force must avoid strongly coupling to protons. Why? To maintain consistency with decades of high-precision physics experiments that have meticulously tested proton interactions and properties. If there were a new force strongly grabbing protons, we almost certainly would have detected major deviations in atomic behavior, nuclear physics, or particle scattering experiments already.
So ignoring protons keeps it hidden from many standard experiments.
Exactly. This very specific selective coupling, primarily targeting electrons via a new fifth force, is what would allow the dark plasma to be precisely confined, guided, and organized by the electrical activity, the Verkage curvatures, generated by our ordinary cells, without disrupting the ordinary matter itself in easily detectable ways. #523 astrophysical and laboratory evidence for the fifth force.
Now, the idea of a fifth force, something connecting the dark and visible worlds, isn't entirely out of left field in cosmology, is it? Astrophysicists have talked about needing something like this.
That's absolutely true. The idea has been gaining traction in astrophysics for a while now, driven by observations that gravity alone can't easily explain. For instance, Antonio Bovea, a physicist involved with the Atlas experiment at CERN, theorized back in 2018 that for the visible and dark universes to have influenced each other's evolution the way they seem to have, some kind of mediating force beyond gravity is likely required. He suggested it might be carried by hypothesized dark bosons.
And there's evidence from how dark matter is distributed.
Yes. Detailed analysis of the distribution of dark matter in galaxies shows that it tends to follow what's called a maximum entropy distribution. That's a state of equilibrium, like how gas spreads out evenly in a container. For dark matter to have reached this smooth equilibrium state so relatively quickly in cosmic history, calculations suggest it must have interacted and exchanged energy, essentially, collided both with its self-interacting dark matter and to some extent with ordinary matter.
Collisions imply a force.
Exactly. It implies an interaction force significantly stronger than gravity must be at work, allowing it to thermalize and reach equilibrium. So there's astrophysical motivation for such an interaction.
But this remains theoretical, maybe even speculative, until we actually find evidence for the force carrier particle itself. And this is where Alfred connects his whole hypothesis to a potentially profound physical anomaly found in an actual laboratory experiment,
Correct.
the proposed discovery of the X-17 particle.
This really is the potential smoking gun for Alfred's coupling model. To understand it, we have to introduce the work of Attila Krasznahorkai and his research group at the Atomkai Institute for Nuclear Research in Hungary. Starting around 2016, they began reporting repeated observations of anomalies in certain types of nuclear decay, anomalies that strongly suggest the brief existence and subsequent decay of a completely new fundamental particle, a new boson, a force carrier with a mass measured consistently around 17 megaelectron volts.
17 MeV, which is relatively light for a fundamental particle, isn't it?
Yes, quite light compared to, say, the W and Z bosons that carry the weak nuclear force. This lightness is important because it implies the force it carries would be relatively short-ranged. And 17 MeV is precisely the kind of mass range you might expect for a particle mediating a new subtle fifth force like the one Alfred needs.
Okay, let's talk about the experiment itself. What were the Atomkai group actually doing, and what was the anomaly they saw?
Their initial, widely reported experiment involved firing high-energy protons at a target made of lithium-7 atoms. This collision momentarily creates an unstable, excited state of a beryllium-8 nucleus.
Okay.
Normally, this excited beryllium-8 decays very quickly, often by emitting pairs of particles, an electron and its antimatter counterpart, a positron. Standard model physics predicts exactly how these electron-positron pairs should emerge specifically. The angles between them should follow a smooth, predictable distribution curve as you vary the energy.
A smooth curve, predictable angles. What did they see instead?
They saw a bump. More electron-positron pairs were showing up at one specific unexpected angle around 140 degrees between the electron and positron paths than were mathematically predicted by the standard model. A statistically significant excess, a spike in the data at that angle.
A bump in the data usually means something unexpected is happening, some unknown process.
Exactly. That excess, that bump, strongly suggested that the excited beryllium-8 wasn't just decaying directly into an electron-positron pair sometimes. Instead, it looked like it was occasionally decaying first into an electron, a positron, and a new unknown intermediate particle. This mystery particle would then immediately decay itself into the electron-positron pair that created the anomalous signal at 140 degrees.
And from that decay, they could calculate the mass of the mystery particle.
Correct. By analyzing the energy and momentum of the electron-positron pairs in that bump, they calculated the mass of this short-lived intermediate particle to be right around 17 megaelectron volts. Hence the name they proposed, X-17.
And they've seen this in other reactions too.
Yes. Importantly, they subsequently reported observing similar anomalies consistent with the decay of a particle at the same 17 MeV mass in nuclear transitions involving helium-4 and carbon-12 nuclei as well. This repetition across different nuclear systems significantly strengthened the case that they were seeing a real particle, not just some experimental artifact in the lithium setup.
Okay, so an anomaly in nuclear physics suggests a new particle, the X-17. What makes this specific particle the perfect candidate, in Alfred's view, to mediate the coupling between dark matter and our bioelectric fields.
This is where the theoretical analysis comes in, particularly from Jonathan Feng and his team at the University of California, Irvine. They took the ATOM Chi experimental results seriously and rigorously analyzed what kind of particle the X-17 would have to be and what its properties would need to be to explain the data within a consistent theoretical framework.
And what did Feng's team conclude?
They concluded that the X-17 is most likely a vector boson. You can think of a vector boson as a fundamental messenger particle that transmits a force, analogous to how the photon transmits the electromagnetic force. But critically, Feng's modeling determined its likely coupling properties, how strongly it talks to other particles.
And its couplings matched the required selectivity.
Perfectly. Their analysis indicated that the X-17 particle couples relatively weakly to ordinary protons, but exhibits a measurably stronger coupling to ordinary neutrons and electrons.
There it is again. That exact selectivity needed. Weak to protons, but stronger coupling to the electron.
It fits the requirements for Alfred's proposed fifth force like a glove. The X-17 being relatively light and therefore mediating a force with a short range, calculated to be around 10 femtometers, roughly the size of a nucleus, would mediate an interaction that primarily attracts dark sector particles, like dark electrons, to the electron clouds of ordinary matter, while mostly ignoring the protons in the nucleus.
How confident were they about this interpretation?
Feng was quoted as stating that, based on their analysis of the ATOM Ki data's consistency and statistical significance, the probability of the Hungarian results being caused by anything other than this new X-17 particle and the associated new fifth force was approximately 1 in a trillion.
1 in a trillion, that's extremely strong wording from a theoretical physicist.
It is. While the existence of X-17 is still debated and requires independent confirmation from other labs, the ATOM Ki results and Feng's analysis provide extremely strong, albeit still technically disputed, experimental grounding for exactly the kind of coupling mechanism Alfred's hypothesis requires to link dark plasma to our bioelectric fields.
Yeah.
Hashtag 5.4, a dark matter plasma skin on the bioelectric field.
Okay, so let's try and tie all these threads together now. The measurable electrical shadow from Levin's work, the detailed plasma structure seen by sensitives, and this potential X-17 particle mediating a fifth force. How does Alfred propose this mechanism actually leads to the formation of that invisible sheath or structure?
It builds logically. It starts with the coupling force mediated by X-17. If dark electrons, the negatively charged particles within the stable dark plasma that Alfred hypothesizes exists all around us in a dark plasmasphere, are attracted to ordinary electrons via this short-range X-17 interaction, then they will naturally be drawn towards areas where ordinary electron density is highest, or perhaps more accurately, to regions where there is significant voltage curvature.
Voltage curvature. We need to define that clearly. What does that mean in this context?
Okay, think of the ordinary bioelectric field around a developing structure, like the tadpole face, as a kind of topographical map. But instead of elevation representing height, it represents electrical potential, or voltage. Voltage curvature is essentially the measure of how sharply that electrical landscape is bending or changing shape across space. It's the spatial gradient of the electric potential gradient.
So areas where the voltage changes rapidly over a short distance, like the edges of the electrical shadow.
Exactly. The electrical shadow seen by Levin, that blueprint of the future mouth or eye, is fundamentally a spatially organized region of high voltage curvature. It's where the electrical potential map is highly structured and changing rapidly.
And this region of high curvature acts like a magnet for the dark electrons, thanks to X-17.
Precisely. The coherent, ordinary bioelectric field, particularly these regions of high curvature, acts as a highly organized field lens. This lens, through the X-17-mediated fifth force, attracts and focuses free-floating dark electrons from the surrounding dispersed dark plasmasphere into these specific zones. Okay. This process creates a targeted influx of dark charged particles flowing towards the blueprint. Alfred calls this flow a dark current, or perhaps a dark electron drift.
So dark electrons start to cluster around the ordinary electrical blueprint. What happens then?
Once these dark electrons cluster in significant numbers around the high curvature regions of the ordinary bioelectric field, they naturally generate a localized region of high negative dark charge density.
A pocket of negative charge in the dark sector.
Yes. And this high negative density then utilizes the dark sector's own internal electromagnetic force, that weak dark electric charge we talked about, to electrostatically attract positively charged dark particles, dark protons or dark ions, from the surrounding dark environment.
Attracting the positive counterparts to neutralize.
Exactly. This leads to local charge neutralization within the dark sector itself, right around the ordinary blueprint. And when you have a collection of interacting positive and negative charged particles that becomes overall neutral, you get the characteristic, highly organized collective behavior of a plasma.
Ah, so the clustering leads to plasma formation in the dark sector.
Yes. The physical result, according to Alfred's model, is the formation of a structured, self-organizing dark matter plasma sheath or envelope that essentially condenses onto and is shaped by the ordinary bioelectric field structure.
And this sheath is the invisible structure, the scaffolding that the sensitives are reporting they can see.
That's the hypothesis. This dark plasma sheath becomes the skin of the ordinary bioelectric field. It dynamically mirrors the shape and changes of the bioelectric template, providing a persistent physical structure for what Burr observed electrically as the blueprint and what sensitives describe visually with color and form. And crucially, Alfred proposes this sheath acts as the primary interface, the bridge, linking the ordinary matter body to a much larger, more complex, theorized dark matter plasma body associated with the organism.
And this model also offers a solution to that computational problem observed by Levin, the mind-boggling computations needed for development.
It potentially does, yes. While Levin observed the need for these computations in bioelectric memory access, Alfred argues that the ordinary bioelectric field alone, based on proton currents in salty water, might be insufficient for the speed, complexity, and robustness of calculation and memory storage required for morphogenesis. He proposes that the dark plasma sheath augments the ordinary bioelectric field, providing additional computational and memory functionalities. It could act like a distributed biological hard drive and parallel processor, operating using the dark plasma dynamics right alongside the ordinary biology during development and regeneration.
So the dark plasma component handles some of the heavy lifting for computation and memory.
That's the idea.
Yeah.
This could help explain morphogenetic robustness, how organisms often correct for developmental errors, the existence of field-based memory, and the kind of non-local biological coordination that seems necessary for organizing trillions of cells into a coherent organism.
And the final piece of the puzzle, the dark site reported by the sensitives. How does the model account for that?
The model suggests that the continuous feedback loops established between the ordinary bioelectric system and this coupled dark plasma sheath could facilitate the development or activation of dark sensory modalities. This might include dark sight, a biological capacity to directly perceive the low-energy dark matter particles comprising the sheath and perhaps the larger dark body.
So perceiving the dark matter directly.
Potentially. Alfred speculates that the activation of these unique perceptual functions might be genetically predisposed in some individuals, explaining why only some people report these abilities, or perhaps it could even be something that can be developed through rigorous training, as suggested by some traditions. The key point is that Alfred's model provides, for the first time, a structured, physics-based framework invoking dark matter, fifth forces, plasma physics for potentially understanding phenomena that have previously been relegated solely to the realm of subjective, anecdotal perception. #tag conclusion.
Wow. This has been a truly monumental deep dive, unpacking the intricate work of independent researcher J. Alfred. We started with the bedrock, the measurable facts, the proton currents in our cells creating the electrical foundation of life.
The ordinary bioelectric field.
We confirmed the reality of that electrical shadow, the blueprint seen by instruments like Michael Levin's voltage dies.
Right, the pre-pattern.
We then explored the historical and experimental evidence suggesting a strong correlation between these measurable patterns and the persistent, detailed, seemingly plasma-like structures visually observed by sensitives over decades.
The gap between measurement and observation.
And finally, we arrived at the core of Alfred's hypothesis, the idea that the potentially real X17 particle acts as a messenger for a new fifth force, a force that selectively attracts stable, self-interacting dark plasma to areas of electrical curvature in our bodies, thereby forming an organizing sheath, a skin, around our ordinary electrical blueprints.
It's a comprehensive framework, certainly. And by positing this stable, self-interacting dark plasma sheath, the model offers a novel physical basis for potentially explaining a range of biological phenomena, things like rapid regeneration, field-based memory storage, non-local coordination during development phenomena that often seem to defy easy explanation by the ordinary biochemical model alone.
It provides a testable hypothesis, doesn't it? Though the tests are challenging, involving particle physics and potentially new detection methods, it's ultimately dependent on confirming the existence and properties of things like the X17 particle and its very specific coupling strengths.
Absolutely. It connects observations from biology, parapsychology, and medicine to cutting-edge theoretical and experimental particle physics.
This integration, particularly of particle physics and developmental biology, it really does open up genuinely uncharted territory for research, doesn't it? The paper suggests not only that our body might be regulated by two distinct forms of electromagnetism operating simultaneously, our familiar ordinary one and a hidden dark one, but that there might even be a kind of intersector co-evolution between ordinary matter life and the dark sector that shapes the form and function of life as we know it.
Indeed. And that leads to some profound final thoughts. If the core computational power, the long-term memory storage, and perhaps the ultimate instruction set for development, regeneration, and repair are stored, at least partly, in a non-ordinary, stable, dark, plasma-based sheath coupled to our biology.
Yeah.
The implications for understanding biological robustness, resilience, maybe even aspects of consciousness, and what constitutes life itself are quite profound.
Yeah.
It raises an ultimate question, really.
Which is?
If the necessary stability and organizational structure for complex information processing can be achieved using something like a weak dark charge and a neutral particle buffer within the dark sector itself, what are the limits of that self-organization? Could life, perhaps in a form utterly alien to our biochemistry, potentially exist and evolve entirely within the dark sector, using its own physics? Something for you, our listeners, to mull over as you integrate this rather challenging but fascinating perspective.